to be affected by these large disease outbreaks that we're seeing on the reef. And if you're not familiar with what a brooding coral is, it's a coral that has internal fertilization. So the sperm are released, captured by another colony, and then the larvae are internally fertilized, and a fully developed planula larvae is released. So a lot of this internal fertilization mechanisms are still unknown in these corals because it's all internal. So I chose to study Parietes asteroides, which is a very common coral on our Southeast Florida reef systems. So the three main objectives I chose to get at were: how does colony size and density influence paternity? How many sires fertilize eggs of one colony? And does this paternity share change over multiple day spawning period? And then, do these corals have the ability to self-fertilize? So my experimental setup, I had 63 corals, and they were isolated, so that I put them all in a sand patch because I was doing genetics on these corals. So I didn't want any other genetics from any other corals in the surrounding area to influence mine. So I had 3 different densities of corals, one at high density, which is 1 meter separation, one at moderate, which was 7 meters in separation, and one at low density, which was 15 meters in separation. And I replicated this 3 times, and all of the replication was split 30 meters apart. We put these corals out on the reef in March of 2016, and then during their spawning period in May of 2016, we brought them back to our lab at Nova Southeastern University, where we spawned them in buckets. It's very important that I spawn them in buckets because then I know those larvae were produced from that maternal colony when I went and did the genetics. So 24 of my 63 colonies actually released larvae, and 13 of those colonies released larvae over multiple days, resulting in 3,184 larvae that I had collected in the 4-day period. So the next step was to do genetics on all of the colonies. So I did genetics on all 63 adult colonies and about 300 of the larvae. I didn't have the time or money to do 3,000 larvae for a master's thesis. So this is an example of what the genetics looks like. So on the left-hand side, you'll see maternal colony ID. So this is colony number 449. And across the top are 7 microsatellite loci that were specifically designed for Parietes asteroides. And then underneath each of those loci, you'll see 2 alleles that are found with that loci. So if you're looking at PA3, the alleles for this maternal colony are 343 and 363, meaning it's heterozygous. But if you look at PA69, they're both 190, meaning that allele was homozygous. Now, if you look at the 15 resulting larvae here, you'll see that for PA3, they all had 343 and 363. And then if you look at PA69, they all had 190. And this trend follows across all the larvae with the maternal colony. So all the larvae in this case were parthenogenic. And this happened for all of the colonies that released larvae. So 100% of the larvae that were released in my study were all parthenogenic. It's a pretty cool finding. This is the first Caribbean coral that's known to reproduce parthenogenically. There's only 2 in the Pacific. So if I didn't convince you enough that this is parthenogenesis, when you normally have DNA crossing over, you have a paternal chromosome and a maternal chromosome. And then DNA is replicated, and you would have some sort of genetic recombination from the crossing over. But in my case, there is no genetic recombination. They are all exact clones of the maternal colony. This can lower genetic diversity in the coral, which can be detrimental in future environmental conditions because it does not allow them to adapt very well to these conditions if they're just cloning themselves. However, it can be good because in situations where there's not a lot of colonies, they don't need males to reproduce. They can just reproduce themselves. So I'd like to thank all my lab members at Nova.